Okay, let's wait for others to join. You had a class now?
No, sir, we didn't.
Okay. Okay. So we'll continue from where we let off last time. I'm trying to call others. Um, okay. All right. Let me share my slides. All right. So we stopped, I think, around this slide. I kind of vaguely remember we started and then I said we will do it in the next class. So we are looking at this class. We are looking at enzymes that have enabled the recombinant DNA technology or genetic engineering to explode. These things we will be discussing on restriction enzymes, and you will realize that something that started as unusual observation, unique observation, just as Alexander Fleming, when he saw that in the near the vicinity of the mold plasmodium, not plasmodium, sorry, penicillium, there was no growth. He could have discarded the plate. He could have just simply discarded the plate, saying, okay, some fungal contamination has happened. But what marks all these remarkable people are that they saw an observation and something stuck within them, and they said, oh, there is something here. If in the presence of fungus, a bacterium is not growing, that means it must be secreting something, or there is something in the fungus that prevents these things from growing, right? And they eventually extracted penicillin, and we all know that the discovery of penicillin heralded the antibiotic era, right? Similarly, restriction enzymes, when they were discovered, it was discovered in a very, very unusual phenomenon. And then when they explored deep down, they realized it was hypothesis. Eventually, that hypothesis was proven, and restriction enzymes came out into the scene, and that heralded the beginning of advances in molecular biology. Okay, so things like this T4 polynucleotide kinase, in the previous thing we talked about terminal deoxynucleotide transferase and few other enzymes. All these things have enabled, what was lab curiosity into actual applications. Okay, so what does T4 polynucleotide kinase do? It basically transfers the gamma phosphate of an ATP to the 5' end of any polynucleotide. It could be, a single-stranded DNA or double-stranded DNA. It prefers double-stranded DNA, but it can still manage single-stranded DNA. Okay, so what is gamma phosphate? If you remember your basic class in GCMB 203, I picked up this from there. Each nucleotide comes in the form of a triphosphate. There are three phosphate groups. Okay, the one that is proximal to the 5' end is called as the alpha phosphate, and the middle one is called as the beta phosphate, and the last one is called as the gamma phosphate. When there is a pyrophosphate release, these two units, the gamma and the beta phosphate gets released, and you have the alpha phosphate. Okay, whenever there is a pyrophosphate release. Okay, otherwise, you know this gamma phosphate is the one that can be readily transferred to other nucleotides. Okay, this enzyme T4 polynucleotide kinase. The reason why it is called as polynucleotide is the substrate. The substrate for this enzyme is a fragment of DNA, which is nothing but a series of nucleotide, polymer of nucleotide. That's what is called as polynucleotide. And as we can understand, a kinase is an enzyme that transfers a phosphate group. Okay, it is not a protein kinase; it is a polynucleotide kinase. We all know, as I said in the last class, we have seen the role of how protein kinases, like CDC kinase, cell division control kinases, have this ability to transition from one phase of cell cycle to different phases of cell cycle. They are also kinases, but they are protein kinases which transfer a phosphate group from from ATP to a protein, right? In this case, the polynucleotide kinase, the substrate is DNA, okay, specifically double-stranded DNA. And as I said, it does work on single-stranded DNA, but with a less efficiency, okay, and transfers the gamma phosphate group, okay. Many a times, you can also have radioisotopes of P32. Okay. In this case, this is ATP. What I'm showing you here is ATP. And what you will see is you can have labels. Either you can have the alpha position of the phosphate labeled with P32 or the gamma position of the nucleotide labeled with P32. Depending on the use, you will either use gamma P32 ATP or alpha P32 ATP. Okay, depends on the use. There are certain applications in molecular biology where gamma P30 ATP is not useful, okay, especially when you're trying to make radioactive probes, okay. In those cases, in certain applications, it's the alpha phosphate. You need to label the this thing. You need to use isotopes that are labeled at the alpha position. And in this case, when you're trying to use T4 polynucleotide kinase, you will need the radioisotope that is gamma, that is labeled at the gamma position. Okay. Can we move to the next slide?
Yes, sir.
Okay. You have another enzyme called alkaline phosphatase. As you know, the kinase and alkaline phosphatase are at extreme diametrically opposite ends. Okay, what alkaline phosphate does is it catalyzes the removal of phosphates from polynucleotides. Okay, so unlike kinase, which transfers a phosphate group, the alkaline phosphatase basically essentially catalyzes the removal of phosphates from polynucleotides, and this also is very, very useful in making sure that when you do cloning reactions, when you want to clone some rare DNA fragments, there are some neat tricks which can use this enzyme alkaline phosphatase to make sure that you have very highly efficient cloning processes. We will probably look in the forthcoming slides how this happens. And sometimes, as we all know, ligation reaction is between a 5' phosphate and a 3' hydroxyl, right? And when you treat the vectors, the plasmid vectors with alkaline phosphatase, what this enzyme does is it removes the 5' phosphate groups from the plasmid. So the ligase cannot join the plasmid. Okay. So whatever colonies you get are going to be recombinant. Okay. Your insert fragment, okay, will give the phosphate group, and the plasmid cannot religate back because you have treated them with alkaline phosphatase. So there is no phosphate group on the vector. Okay, so the phosphate group from the vector is removed because of alkaline phosphatase, and your incoming DNA, the DNA fragment that is of interest in cloning, that provides the phosphate group. And so because of that, the every colony that you're going to see on your plate, is more than 90% of them are going to be recombinant. That means they will have a foreign gene of being getting inserted. The reason I'm saying more than 90% is 10% of the times, there is a small this thing population that may not get dephosphorylated. Okay, so they might come back, but that's a very very rare event. Okay. Okay. People have also used a series of enzymes called nucleases, which you must be familiar right from your senior school. When the people transformed, when how was DNA shown to be the the this thing for inheritance. The molecule responsible for inheriting a property. They simply did a nuclease digestion and they realized that the S pneumonia did not become R pneumonia, right? Or vice versa, right? So it turned out that there are many different nucleases that have this ability to chew up or digest the nucleotides into the individual components. Okay, so very broadly, nucleases can be divided into two classes. One is called as exonucleases, which removes the nucleotides one at a time from the ends of the DNA molecule. It cannot act in the middle. Okay, so it needs to have a free end. And as I re-emphasized last time, if you were to take any exonuclease and incubate with a perfectly circular plasmid DNA, nothing will happen. Okay. On the other hand, if you take a linear DNA, right? For example, when you make a genomic DNA from any organism, right? It could be from E. coli, it could be from any plant sources. And when you add a exonuclease to it, it will start chewing up. Okay. Endonucleases, on the other hand, okay, the distinction between exon and endonucleases is that the endonucleases break the phosphodiester bonds within a DNA molecule. That means, as far as endonucleases are concerned, it just doesn't matter whether you're presenting them with a linear DNA or a circular DNA. Okay, endonucleases recognize certain specific sequences. Okay, and they will go and recognize that and they will cleave. They will remove the phosphodiester, cleave the phosphodiester bond, and that is how a circular DNA will become a linear DNA, or if it's presented with a linear DNA and if the sequence is present, it will go and cut in those in those sites which are called as recognition sites, and typically restriction enzymes are also called as restriction endonucleases. Okay, because they cut right in the middle, they don't start chewing up from the end. Okay, is this distinction between exonuclease and endonuclease clear?
Yes, sir, it's clear.
Okay. We'll quickly look at some of the exonucleases. There is an exonuclease called BAL31. Okay, it's a double-stranded exonuclease, operates in a time-dependent manner, and degrades the DNA from both the 5' and the 3' ends. And this is very, very useful for generating deletion sets. We will, during our class on genomic library construction, we will see what a deletion set means. Okay, and the longer you incubate, you will have a bigger deletion. Okay, the longer you incubate, obviously it is going to chew up more and more DNA. Okay, so just imagine incubating this a piece of DNA. Okay, 50 kilobase pairs of DNA with BAL31 for 5 minutes versus incubating in another parallel reaction for 1 hour. Obviously, in 1 hour, it is probably going to chew up a lot more nucleotide than what it did in 5 minutes, right? So another thing with the difference between a restriction endonuclease, I'm only talking about an endonuclease, okay? The difference between a restriction endonuclease and an exonuclease is that exonucleases will keep on chewing until the DNA has come into single polynucleotides, into single nucleotides, right? On the other hand, restriction endonucleases, when they recognize a certain restriction site, they will cleave it, and then that's the end of it. The DNA doesn't get degraded. Okay, so that's a big difference between exonuclease and endonucleases. So exonucleases have this ability to degrade DNA into its individual nucleotide units. Okay, restriction endonucleases generally do not do that, but there are other types of endonucleases that can act pretty much like exonucleases. That means once it is chewed, it will start chewing up from the other ends also. Okay. Okay. Just as you have BAL31, you have another nuclease called exonuclease 3, okay, which prefers double-stranded DNA, okay. It has a 3 prime 5 prime exonuclease activity, unless BAL31, which can act on both 5 prime and 3 prime end, okay. This has activity from the 3 prime 5 prime exonuclease activity. Some 3 prime overhangs, okay, are resistant to activity, okay, and this property can be used to generate what is called as nested deletions from one end of a piece of DNA. We will see, as I said, we will see some of these examples when we start looking at how to make genomic DNA, how to make sets of genomic DNA libraries. Many a time, some of these enzymes are going to be used in those things. Okay, exonuclease 1, unlike exonuclease 3, exonuclease 1 works on single-stranded DNA. Okay, so this is very useful for removing the primers from a PCR reaction or from other reactions where primers could be used. Okay, so you don't want these primers to be interfering in your cloning purpose or for any other downstream reaction. When you don't want them, you can use exonuclease 1. It will chew it up. Okay, all the primers will be gone. Okay, DNase 1 is one other endonuclease. Okay, this is not an exonuclease. This is an endonuclease. It cleaves double-stranded DNA randomly. Okay, it can also cleave single-stranded DNA, but the preferred substrate is double-stranded DNA. Now, in the presence of manganese, okay, it cleaves both the strands of DNA. It can cut both the strands of DNA. In the presence of magnesium, it creates what are called as single-stranded nicks. Okay, imagine it's a double-stranded DNA. It will randomly create nicks. That means it will remove phosphodiester bonds here and there. Okay, not everywhere, here and but as long as your concentrations are very high, it is probably going to chew up like exonuclease. But when typically when we do these reactions, we include very small amount of. We don't put large excess of enzyme. We put only a minimal amount of enzyme. Okay, with that minimal amount of enzyme, it generally creates random nicks. Okay, and also we leave it for a short time. We don't leave it for 1 hour, 2 hour, and all that. So if you leave it for 1 hour, 2 hour, or longer, it is going to become all these double-stranded DNA will become individual nucleotides. Okay. Now it also depends, as you can see, depends whether you are using magnesium in your buffer or manganese ions in your buffer. When you use manganese ions, it is going to cut both the strands of DNA. And if you don't want it, if you want only single stranded, then you will be using magnesium. Okay. And this enzyme is very useful for defining binding sites for DNA binding proteins. Imagine DNA is something that is always bound by some protein or the other. For example, DNA polymerases are binding to DNA for transcription, for replication. RNA polymerases are binding to the DNA for transcription. Topoisomerases bind to DNA to make sure they are all in a topologically supercoiled state, right? You have many different kinds of proteins, single-stranded DNA binding proteins, right? So many different kinds of proteins that can bind to the DNA. You have transcription factors, right, which can bind to DNA and make sure that the gene is active, or it can also shut down depending on the protein that we are talking about. Suppose you want to know, somebody says, let me know if this is a DNA binding protein or not. There's a very simple assay that can tell you whether it's a DNA binding protein or restriction enzymes are DNA binding proteins, right? On the other hand, Any enzyme that participates in, say, glycolysis or TCA cycle, they are not going to be binding to DNA, right? So you definitely have two distinct class of proteins in the cell, or maybe three distinct class of proteins in the cell. Those that bind to DNA and also are involved in the modulation of gene activity, and those that do not bind to DNA, right? Very broadly. Okay. So we are going to look at how can we know if somebody says, these days, of course, it's very easy, right? All you need to do is when somebody gives you a, protein, if you're able to figure out what this protein is, all you need to do is go back to your bioinformatics tools and figure out whether this is a DNA binding protein or not. If suppose no information is known, and let's just say that the fifth semester students went on a trip to a remote island and found something unusual, and they purified an enzyme or purified protein, and they realized that this could be a DNA binding protein, could be a new restriction enzyme. How do we know whether it's a restriction enzyme or not, or whether it's a DNA binding protein or not? Very, very simple. Okay. All you need to do is take radioactively labeled DNA, double standard DNA. Okay. You need to calibrate. Okay. This DNA is commercially available now. You can get purified DNase one from commercial vendors. You need to use, so that means you're not going to get this experiment right the first time. You probably need to play around with the concentration of DNase one such that it hits one hit per DNA molecule. Suppose you have in this case, for example, 1, 2, 3, 4, 5, 6, 7, 8, 9, 9. This thing. Suppose you take, say, a small amount of DNA. You want to make sure that the DNA nicks at each place only once. Okay, there's going to be a population. There is going to be a population. Okay, in this case, for example, there are some population in which the DNase one nicked here. In some population, it nicked here. In some population, it nicked here. Okay. So you have a series of this thing. So that is what it says. It's very, very crucial to make sure there is only one hit per DNA molecule. So you're going to use an extremely low amount of DNase one. Okay, low amount of DNase one, such that the interaction between the DNA and DNase1 is going to be limited. Okay. And when it is limited, it is so limited that it is going to randomly hit at only one of these spots. Okay. So you are going to take a radioactively labeled DNA. Okay. You will aliquot this into two tubes. Okay. And in one, you are going to bind this protein. Okay. We don't know whether it's a DNA binding protein or not, but let's assume that this is a DNA binding protein, when it binds right in the middle of this. Okay. And then you're going to subject this to DNase one. Okay. When you incubate with DNase one, in this sample, because no protein is bound, it is going to chew up at these different places. Okay. In this case, because the protein is bound in the middle, it will chew up on either side of it, right? And when you separate this on a electrophoresis, gel electrophoresis, then what will happen is, in this case, where there was no protein present, it is going to give you a series of fragments, right? It is going to give a series of fragments which you can visualize on the autoradiogram, right? This is the smallest fragment because it hit right here, okay? And this is going to be the largest fragment here, okay? So it is going to run, your electrophoresis is going to separate them based on the size, right? So this is the least, and so this is going to move faster, and this is the largest fragment. It's going to be migrating slowly. Okay. Now compare this with this. When the protein is bound, okay, because it is not able to generate any nicks here, what you see on this is there is a gap here. Okay. And then people now know that it is this region of DNA to which this specific protein binds to it. Okay, so that is how people know whether it's a DNA binding protein or not. And then, you have a series of reactions in which you can, do a sequencing reaction and figure out what is the sequence to which this DNA protein, this DNA binding protein binds. Is it non-specific or is it specific, right? So you can figure that out. So this is just showing an example from a research paper when this technique was first introduced, okay? So Drosophila heat shock factor, okay, heat shock protein goes and binds very specifically to this region, okay? This is the different concentrations of DNase 1, okay? At this concentration, you can actually see that, the DNA binding protein has bound to this region and it protects. This is why it is called as DNase protection assay, and it is also called as DNase 1 footprinting. Both mean the same thing, DNase 1 protection assay or DNase 1 footprinting. Okay, so this is what is called as the footprint. Okay, these are the places where the heat shock factor is interacting with the DNA. Okay, did I make it clear or should I explain it?
Sir Rook, if you could explain it again.
Okay. So we'll go back to the previous slide, right? So this is the labeled DNA. Okay, it has been labeled at one end with the radioisotope. Okay, it could be P32 ATP, P32 GTP, anything. It is labeled on one end with the isotope label. Okay, you would divide this into two aliquots. Okay, one in which you are going to use extremely low amounts of this enzyme called DNase 1. The reason why I said, you need to calibrate the nicking at one hit per DNA molecule is because you need to, experiment different concentrations of DNase 1. Okay. When you get a vial of DNase 1, you will not know how much DNase 1 to use. So you may have to dilute, maybe 1 to 10 dilution, 1 to 20 dilution, 1 to 50 dilution, or maybe 1 to 100 dilutions, and check, this process where it is going to hit only once. Okay, so you're going to use an extremely diluted DNase 1, okay, so that it introduces only one nick. Otherwise, what will happen is you will not be able to have this kind of a ladder pattern. Okay, so now you do this reaction of with DNase 1 in two aliquots, one in which your DNA is alone, and one in which the specific protein has bound to the DNA, right? You subject both of them to DNase 1 nicks, and in this case, it is going to nick at every possible place, okay, randomly at every possible place. In this case, typically what happens is wherever the protein is bound to the DNA, DNase 1 cannot go and introduce a nick, cannot degrade at those places, right? So typically what happens is, in this case, when DNase 1 nicks at literally every single nucleotide, you're going to generate what is called as a ladder. Okay. So this is the shortest DNA fragment from the label. What are you detecting on the autoradiogram? The radioactively labeled ends, right? So in this case, this is the shortest one. So this is going to migrate the fastest, and this is the longest one, so it is going to migrate the slowest. Okay, now what you see is a continuous ladder pattern, right? In this case, when the DNA has been bound by a DNA binding protein, and when you do the same reaction with DNase 1, it nicks at these places, but this region is protected by the DNA binding protein, right? The DNA binding protein and DNase 1 can't be acting together. It's like something is stuck, and so DNase is not able to go and introduce a nick in those sites. Okay, so when you run both the samples on the electrophoresis, so this is called as polyacrylamide gel electrophoresis, or when you do a page, typically this is the pattern that you see. Okay, so what you see here is a region that is missing. Okay, and this is the region where your DNA, the specific DNA is bound, and so this assay is also called as DNase protection assay or DNase 1 footprinting assay, very popularly called as DNase 1 footprinting, but it is also called as a DNase protection assay. Basically this region is protected from DNase 1. So people also called as DNase protection assay. But I think DNase 1 footprinting seems to have been more popular than DNase protection assay. Okay. So when you run, then what you see is a short region in the autoradiogram, which is you don't see the ladder pattern. Okay. So based on the sequencing reaction, you will be able to know what is there a preference for this protein to bind to certain sequences, or does it bind randomly at any other place? Okay. So this, just I'm showing you a research article in which Drosophila heat shock factor, okay, this is the first paper actually that showed the utility of this. So I'm putting out a historic data, okay. So heat shock factor at increasing concentrations of this, heat shock factor, you can actually see that this is the region of DNA that is protected from DNase 1 when heat shock factor binds to the DNA. Okay. You also run a parallel sequencing reaction, so you will know, what is the base pairs to which this DNA protein binds. In this case, heat shock factor has certain recognition motif. It doesn't go and bind to everywhere in the genomic DNA. It goes and binds to very specific region. So you will get to know, what region it is binding to. Okay. Can we go to the next slide?
Yes, sir.
Okay, thank you. Okay, so we look at topoisomerase. This is something that I briefly alluded to when we talked about in the cloning part. Okay, so this is an unusual enzyme. This is an enzyme that is actually responsible for supercoiling, which is also very, very important because if you remember our class on GCMB 203, basics of molecular biology, E. coli's dimension is so low that if the entire E. coli genome, E. coli DNA has to be fitted into the E. coli's cell, it needs to be supercoiled. And topoisomerase 1 is the one that is responsible for supercoiling of the DNA. But this is also an unusual enzyme that it is an enzyme that can nick the double-stranded DNA, and it can also ligate it back. Okay, it can also ligate it back. Okay, and this is essentially it's going to alter the linking number in a supercoiled DNA. Okay, and this plays a very, very important role. If you remember the basic class in DNA replication, you always have topoisomerases moving not only forward but also behind the replication fork. In front of the replication fork, it relaxes the DNA, and behind the replication fork, it increases the linking this thing, and it twists the DNA back into the supercoiled shape. So this is just showing you, model of how this. So this is your protein. This is the topoisomerase protein, and this is the DNA. Okay. And as it moves, there is a short movie. I'm not showing the movie. Okay, this is just a model to show how this thing. So this is the DNA. The green piece is the DNA. Okay, the green piece is the DNA, and this the blue one is the protein. Okay, and so initial cloning experiments used to use the T4 DNA ligase. Okay, but somebody smart enough said, if topoisomerase can do this function, can I not use topoisomerase for cloning? Somebody did that, and they made a lot of money out of it. They made lots of money. A company started with this system, and they have now patented this system, and they have minted a lot of money. So that's what I keep telling you. This is what we are going to see in our IPR class. In your next semester, that every knowledge that you gain from any system can be monetized. Okay, should it be monetized is a different question. Can be monetized, and this is especially something that they lay emphasis on in many of the universities abroad. Okay, slowly that is getting caught in India also. I mean, we are also into patenting, as you all must be knowing. Amity was conferred with the intellectual property rights. This thing last year, 2020's IP management came to Amity. This was recognized by the union government. So that means Amity has done a fairly good job. Has done a really wonderful job in patenting. Okay, so that culture is coming. So any new knowledge that we gain can be patented, and as long as somebody is seeking an application, people will charge a fees for it. So these kits come at a price. Okay, what used to take overnight ligation, now you can do it in 5 minutes. That means in a matter of maybe by end of the day, you are ready to generate clones for your DNA fragment. That means your what used to take years became months, months became weeks, weeks have now become days. Okay, so that is how does this work? You basically have, these topoisomerase recognition sites, right? And you come with the PCR product, okay? And in a matter of 5 minutes, these PCR product and this, they are ligated by the topoisomerase, and your ligation gets completed in less than 5 minutes. So that is the beauty. Okay, it's extremely efficient. More than 85% of the resulting plasmids are recombinant. So this is an excellent one for making libraries. Okay, why do we even need to make libraries? We will come back. We'll come back on that this thing. Okay, this can be used to clone blunt-ended DNA. For example, cloning a blunt-ended DNA is typically a lengthy process, time-consuming process, less efficient process. But with topoisomerase, this can be done very quickly. Okay, you can virtually do cloning of any DNA, any foreign DNA into a vector. Okay, unfortunately, only thing is you have to use their plasmid vectors. The company that gives this product, you need to use their vector. Okay, so you can't have your favorite. Many people who do molecular biology end up having their own favorite cloning vector. So you can't use your cloning vector. You need to necessarily use their vector. Okay, that's not a big deal. Okay, it's not a big deal. Okay, so we'll quickly get into how cutting and pasting of DNA happens. This is essentially a revisit of the cloning part. So we'll quickly go into this. Okay. How did they discover restriction enzyme? As I said, the entire process of discovering restriction enzyme turned out from a very teeny tiny observations. Okay, somebody who kept on using bacteriophages for infecting E. coli and trying to understand how E. coli gets infected, how the bacteriophage takes control of the host system, what all it does to the host. Somebody who was studying this found a very minute observation. Okay, from that observation came a hypothesis, and that hypothesis was validated. And once it was validated, it had a huge application in molecular biology. And all those people who made the hypothesis, who tested the hypothesis and applied it, all three of them got Nobel Prize. Okay, so this is something that we need to be very aware. Any small observation can have a large implication. Okay, so very quickly, how did they go about discovering this restriction enzymes? So they use two different kinds of E. coli. Okay, you have different strains of E. coli. Okay, they use two different strains of E. coli. One is called as E. coli K, and the other is called as E. coli C. Okay, so bacteriophages that are typically grown in E. coli K. Okay, when you plate them back on E. coli K, they have what is called as EOP of 1. EOP is basically efficiency of plating, which is a measure of phage virulence. Okay, anything that is grown on E. coli K grows very well in E. coli K and has an EOP of 1. Okay, when you take this and when you infect E. coli C, you didn't have any issues. It came back. It came back and it plated very well. Okay. So E. coli K to E. coli C, there was no issue. Okay. When you take these things which are grown in E. coli C and when you try to plate again in E. coli K, they realized that the efficiency of plating had fallen down. It had fallen down so dramatically. It looked like as though these guys have now become resistant to the phages. Okay, although there is a small population, okay, there's a small population that was still susceptible, but by and large, it looked like suddenly E. coli K has become resistant to bacteriophages. Okay, when they realized why it is so, and how it is so, if you read this, if you read this, you will know this. Okay, so I'm leaving at that. Okay. And then they realized that restriction enzymes can have profound applications in molecular biology. Okay, so that is what resulted in the discovery of the endonucleases. Okay, when they discovered it, luckily for them, they discovered this type 2 endonuclease. Okay, but then further research people realized there are at least 4 different types of restriction endonucleases, at least 4 different types. Okay, so the type 1, it cleaves at sites remote from the recognition site. That means it binds at one place and it goes and cleaves at a distance, at a far off distance. Okay, and it requires both ATP and S-adenosyl L-methionine to function. These are typically multifunctional proteins with both restriction and methylase activities in the same subunit. Okay, these are multi-subunit proteins that function in a single protein complex. That means there are multiple activities associated with this enzyme. Remember your old classes in enzyme when people talk one enzyme, one substrate. The enzyme is supposed to have only one function, but this is a multi-protein, multi-complex this thing. So it has multiple activities. Okay, it usually has two restriction units and two methylase units. Okay, type 2 enzyme which they cloned. Okay, recognizes specific DNA sequences and cleaves at constant position which is very close to that sequence. Okay, which results in the production of 5' phosphate and 3' hydroxyls, and this is what is most useful in cloning. Okay, type 3, it is composed of 2 genes which encode protein subunits that function either in the DNA recognition or modification. This is a variation of this. This has not been very useful. And type 4 is again a modification of type 3, but it can recognize methylated, hydroxymethylated, or glucosyl hydroxymethylated bases. So many of these things, when there is a change in the base structure, many a times, as we say, the bases can get different groups attached to them. And when those different groups get attached to them, they become resistant to these endonucleases. Okay. So, but still, type 4 can recognize hydroxymethylated or glucosyl hydroxymethylated or methylated sequences. Okay. Typically, when there is a methylation, then the DNA becomes resistant. It cannot be degraded by, it cannot be, recognized by the restriction endonuclease. Just having one small methyl group, can prevent the restriction enzymes from functioning. Okay, all it needs is a small modification. That means these restriction enzymes are highly, specific for the geometrical structure of the DNA. Okay. Now this is typically restriction enzymes. I'm just showing an example of type 2 restriction enzyme. Okay, they come as dimers. That means 2 subunits will come together, right? One subunit recognizes the bottom strand, and the other subunit recognizes the top strand, right? In this case, the recognition site for EcoR1 is GAATTC, right? From the 5' GAATTC, and these invariably are palindromes, right? I'm sure you will be reading in Fundamentals of Recombinant DNA Technology this in further details. Okay, and so EcoR1 recognizes this specific sequence. So wherever GAATTC is found, okay, E carbon will go bind there and it will cleave right after the first G, okay, on the 5' position. Okay. So at the end of it, this is how the reaction will look like. You have 5' G with a 3' hydroxyl in this, and then you have 5' which starts at AATC and ends with a 3' overhang. Is this clear? Can we go to the next slide? Okay, so these are the example of recognition sequences for restriction endonucleases. Okay, some restriction endonucleases recognize 4 base pairs. Okay, for example, in this case, you know, this enzyme called ALU1 recognizes a sequence called AGCT and cuts right in the middle, and that it generates blunt end. Okay, you have another enzyme called MSP1, okay, that recognizes at the this sequence CCGG and cuts right after the 5' C, the first C, okay, and that it generates a 5' overhang, okay, 5' overhang, okay. You have 6 base pair cutters, which are more frequent. You have PBU2, which again cuts right in the middle and generates a blunt end, okay. Then you have an enzyme called KPN1, okay, which recognizes this sequence GGTACC, and unlike ECOR1, which cuts at this G, it cuts at the end. Okay, so it generally creates what is called as a 3' overhang, and then you have very rare 8 base pair cutters. For example, there is an enzyme called NOT1, which recognizes this sequence GCGGCCGC, and it cuts after the second nucleotide. That means after G and C, it cuts here and results in a 5' overhang. And then you have some unusual enzymes, restriction enzymes, okay, which can have little non-specificity. Only thing is, it needs to have the specificity. Any GC sequence which is surrounded by any other nucleotide. N means any nucleotide. It could be a series stretches of G, it could be stretches of C, it could be stretches of A or T. Only thing is it needs to be surrounded by this GC. Okay. And typically this enzyme results in the formation of a 3' overhang. So you can imagine restriction enzymes or restriction endonucleases come in different forms and flavors. As of now, I think there are close to 4,000 different restriction enzymes that have been cloned. Many of them end up having the same recognition sequence. Okay, one organization that has commercialized this is a company called New England Biolabs. Okay, they are very famous for making many different kinds of restriction enzymes. Okay, and one of the Nobel laureate who discovered restriction enzyme worked with New England Biolabs. Okay, so okay. So how often, suppose I give you, a stretch of sequence and say, find out how often the enzyme will cut your DNA. There are simple, mathematical formula. For a 4 base pair cutter, the site, assuming that your GATP nucleotides are distributed randomly, then for a 4 base pair cutter, it is 4 to the power 4, which means every 256 base pairs, theoretically, every 256 base pairs, assuming random distribution of GATC, you are supposed to have restriction enzyme site. Okay, if it's a 6 base pair cutter, then it becomes 4 to the power 6, which means every approximately 4 kilobase pair. Okay, if it's an 8 base pair cutter, what what you see is every 65 kilobase pair, you are supposed to have one restriction enzyme site for that particular enzyme. This is under the assumption that GATC is distributed equally. But we all know that it is probably not an equal distribution, because I keep telling you the examples of Mycobacterium tuberculosis, which is highly GC-rich organism, and Plasmodium falciparum, which is highly AT-rich organism. So there are organisms whose genomes are biased. Some have GC bias, some have AT bias, and most organisms don't have a bias. But sometimes some of these organisms can have extreme compositions. Okay, so the sequence context makes a lot of has a lot of influence on how this restriction enzymes act. And also, as I said, all it needs is a small methyl group attached to it. The restriction enzymes can be rendered ineffective. Okay. So we are not going to go much into restriction enzymes at this stage. Maybe if time permits, then we will come back and see a short video from the New England Biolabs website, how you could purify restriction enzymes, how you will characterize restriction enzymes, how they commercialize restriction enzymes. I'll probably show some figures. They will also tell you each restriction enzyme, for example, comes with a data sheet. It will also tell you what is the recognition site for it, what is the optimal temperature for it. By and large, when we talk about enzymes, any idea what is the optimal temperature in which enzymes act? Any guesses? What will be the optimal temperature for an enzyme to act? Any guesses? Yes? Take a guess.
So won't it be the let's say for an enzyme that's in the human body, won't it be the typical temperature of a human body? So what 38 degrees Celsius?
Very
That's
good.
my
Yeah, you are right. You are right. So most enzymes perform optimally at 37 degrees centigrade, right? But we also know that organisms not all live at 37 degrees centigrade. For example, the reason why we use stacked DNA polymerase is because it is isolated from a hydrothermal vent where organisms are literally at real near boiling temperature, right? So Taq polymerase works optimally at around 70 degrees centigrade, but it can withstand, few more these things, right? So most restriction enzymes work optimally at 37 degrees centigrade, but the organisms from which Taq polymerase is isolated also has a restriction enzyme called as TAC1, okay? And TAC1 works optimally at 65 degrees centigrade. Okay, and there are certain organisms that grow in cold temperatures, right? They grow at extremely cold temperatures, and their enzyme, when you isolate restriction enzyme from those organisms, they end up working at a lower temperature. For example, there are some restriction enzymes that works optimally at 25 degrees centigrade. Okay, so each, when you buy a restriction enzyme, the restriction enzyme comes with the data sheet, which tells you what is the composition of the buffer, and what is the optimal temperature at which you can do this restriction enzyme. And suppose at the end of restriction enzyme, many a times these can affect your ligation. So you want to heat inactivate, for example, you want to heat inactivate. Can you heat inactivate this restriction enzyme or not? And when you do heat inactivation, typically we all know from our biochemistry class that when you heat any protein, the structure is gone, the secondary structure, tertiary structure, they're all gone, and the enzyme loses activity. One of the easiest way for to kill an enzyme is to expose it to high temperature. So when you do this to restriction enzyme, Will it still be acting or will it be dead? Some restriction enzymes are heat labile. That means they will all die. On the other hand, some restriction enzymes are resistant to heat. It doesn't matter to them. Even if you incubate them at 75 degree or 80 degree centigrade, after a while they will retain some residual activity. Okay, and this is a problem because when you do your ligation, you're going to recreate the restrictions site, and this enzyme can go and cleave it again. So you're going to have an issue. So you need to make sure that at the end of restriction digestion, either you need to remove that restriction enzyme or you need to heat inactivate. Okay. So we will start looking at, I think, this is what I wanted to tell you, that ligation is something that looks very simple, but there is a complex this thing behind it. And the reason why topoisomerase became so popular was because of certain limitations with normal ligation reaction, which we'll probably look it up on Monday. Okay. Any questions at this point in time?
No, sir.
Okay. Okay. So we'll see again on Monday then. All right. So take care.